This is the second part of the cellular energy dynamics PowerPoint. So I'm going to talk about enzymes. In the first part of this PowerPoint, we discussed chemical reactions and the ATP cycle. So just a quick review, you need to know about endergonic and exergonic reactions. Remember, endergonic requires energy, exergonic releases energy. And then you need to be able to apply that concept to the ATP cycle. So ATP loses a phosphate and becomes a DP. Since a bond is broken, energy is released. Now, when ADP gains a phosphate to become ATP, that requires energy. So now we are going to move on and learn about enzymes. So you will need to be able to define activation energy. And also you need to be able to describe how enzymes function in a very general sense. Here is a video that you can view on your own. Now, I do not go all the way through this video. I stop when it starts discussing coenzymes. So let's talk about the function of an enzyme. Now, hopefully you recall that an enzyme is a protein. And this is one of the major functions of proteins. So an enzyme catalyzes and speeds up a chemical reaction. Now remember that catalyzes means it begins the reaction. It helps get it started. So I'm going to write this down here. Catalyzes and speeds up chemical reactions. So most chemical reactions will occur on their own, given enough time. But it takes so long for these reactions to occur on their own that we would not be able to function as an organism just relying on reactions without enzymes. So enzymes speed up those reactions so that they occur much faster in an organism. So the question that I put on this slide is, what is a substrate? So a substrate is the molecule or multiple molecules that fit into the enzyme. So here in this slide, you can see I wrote substrate right here. And here is your enzyme. Now the substrate fits into what we call the active site. Now, this is almost like a lock and key relationship. So a specific substrate works with a specific enzyme. So I'm going to go to the whiteboard to write down a few basic pieces of information about enzymes. Okay, so we're discussing enzymes. Okay, so they are proteins. They catalyze and speed up chemical reactions. An enzyme has what we call specificity. Hard to say. And what that means is a certain enzyme only works with a few given substrates. As an example, there is an enzyme called catalase. Remember that the names of enzymes end in ASE. So the enzyme catalase breaks down hydrogen peroxide, H2O2. So that hydrogen peroxide molecule, H2O2, fits into catalase, but it would not fit into other types of enzymes like amylase or lactase. So for example, lactase only breaks down lactose. Now, an enzyme can also put two molecules together in an endergonic reaction as well. But we mostly talk about enzyme function referring to exergonic reactions. Now, another thing that you need to know is enzymes are not used up during a reaction. They speed up the reaction, the substrate leaves, and the enzyme is free to catalyze and speed up more reactions. So enzymes are not used up during a chemical reaction. Now, how does an enzyme function? Well, what you need to know overall is that enzymes lower the activation energy of a reaction. I'm going to go into this a little bit more in the next few slides. But let's look at exactly how this works. So you're going to have to bear with me on my drawings here. Not very good drawing on the whiteboard, but I think you'll get the general idea. So here I have my enzyme. My pen's not working super well. Okay, so let's say this enzyme is lactase, and I'll add a few labels here in just a second. So this would be my substrate. Now notice it would fit right into lactase like a lock and key. Let's add a few labels. Here we have lactase. That is the enzyme. Here we have lactose. That is the substrate that fits into this particular enzyme. Now, I want you to take a second and think back to our biological molecule PowerPoint. What type of molecule is lactose? Is it a protein, carbohydrate, lipid, or nucleic acid? So you should pause here and think about that question. So lactose is a carbohydrate. Now remember, it's a disaccharide. There are two simple sugars that bond together to make lactose. But in this particular reaction, the lactose is going into the enzyme and it will be broken apart into those two simple sugars or monosaccharides. So what you're looking at here is an exergonic reaction. So I'm going to draw a little arrow here. This is to symbolize the reaction. Okay, so we still have our enzyme. It is not changed by the reaction. But now, instead of a lactose, I don't really know how to draw these. They're not going to have the same shape, so we'll just kind of do this. Now we have two molecules because lactose was broken into the monosaccharides that made it up. So let me label this for you. So we still have lactase, our enzyme. Remember, enzyme names end in ASE. And actually, sugars generally end in OSE. Glucose, galactose, lactose. So lactose was broken up, and now we have glucose Didn't work. Glucose, and you should know already what the other one is, galactose. Now, left on its own, lactose would break down into glucose and galactose, but it would take a really long time. And you need to be able to break down that lactose in your body, right? So people who are lactose intolerant do not have enough of this enzyme. And you can actually get medication that will add lactase to your body so that you can have dairy products. So let's go back to the PowerPoint. Okay, now remember that in my example, lactose would be the substrate. It's actually, if you think about writing out a chemical reaction, the substrate is the reactant, right? It goes on the left side of the equation. Now the substrate fits into the active site. Now what it says here is the enzyme changes shape slightly as the substrate binds. All that means is that the enzyme tightens around that substrate. So why does that happen? How does an enzyme lower activation energy? Well, let's go on to the next slide. This is my image that I liked to illustrate activation energy. So activation energy is the initial energy that is needed to allow a chemical reaction to occur. And I'm going to show you a graph that illustrates activation energy. So if you look at my picture here, the reactants are all these little frogs. Now, activation energy is like a barrier to getting that reaction started. And if it's really high, if you need a lot of energy, that reaction doesn't go very much, right? It never occurs. Like it will occur a little bit, but it will take a long time for the reaction to occur as much as you need to. So you could picture these frogs as lactose substrates, right? If there's no enzyme, it takes a lot of energy to break that lactose into glucose and galactose. Now let's look at the picture with the enzyme. The activation energy, the barrier, is lowered. So more frogs can jump over. To apply that to our chemical reaction, once that activation energy is lowered, there's many more lactose molecules that can be broken down. So remember that enzymes lower activation energy. So let's look at this concept in a graph. Now, this graph is not quite correct. I need to find a better one, but I haven't found one that illustrates what I want you to see very well. So here it shows two molecules as the reactants and one molecule as a product. Oh, actually, this graph is correct. I forgot that I changed it up. Actually, no, it is not correct. Sorry to keep going back and forth. So if you remember, this graph shows you an exergonic reaction, right? So let's look at the y-axis. Remember, I called it potential energy before, right? You can also think of it as energy released, energy absorbed. So let's say that this reactant is our lactose. Now, remember, in an exergonic reaction, you are giving off energy. Bonds are being broken. So you're going from a large molecule to small molecules. That's why I say this graph is not correct, because it shows small to large. So lactose is the larger molecule. We'll just say large. What you end up with, we're going to cross that out, would be glucose and galactose, right? They are small molecules. You've broken lactose apart. All right, glucose plus the lactose. These are small molecules. They do not have a lot of potential energy because they don't have as many bonds. So when you break up lactose into those two monosaccharides, you're giving off energy. So now let's apply this to activation energy. Now look at this, whoops, peak in the graph, right? Okay, my pen is giving me some issues here. So let's print that again. Okay, so we're looking at this green line, okay? Now notice that it peaks and then it goes down. This peak right here, from here to here, is the energy that is required to get that reaction started. Now, this is actually a pretty complicated process, and I'm not going to go into details because you don't need them. But the activation energy is what's needed to start those bonds breaking. Okay, you have to put a little energy in to start breaking those bonds. Now, once you've done that, energy is released because it's exergonic. Now, without an enzyme, energy from the environment in your body, like a cell in your body, right? That energy would have to come from somewhere in that cell. It would take a long time to accumulate enough energy to get that reaction started. This is where an enzyme comes in. This purple line right here is the activation energy once an enzyme is involved. Notice how much, how that there's less energy required. Okay, so the enzyme is lowering that energy that is needed to get that reaction started, to start breaking those bonds. So how does it do that? Well, there's many ways, but I'm just going to give you one example to kind of help you visualize this process. So when a substrate enters an enzyme, the enzyme puts pressure on those bonds. It bends the molecule a little bit, so it's easier to break those bonds. An example I can think of is, let's say you put like a plywood board in between two grips. Okay, if those grips bend the board a little bit, is much easier to break it than if it was just straight. I'm using that because of like martial arts, right? Breaking boards, so much easier if you like bend the board a little bit. Well, that's essentially what an enzyme does to chemical bonds. Puts stress on them, weakens them. So it takes less energy to break them. Okay, I'm trying to advance this. Doesn't seem to want to. Okay, so that's actually my last slide for the enzyme portion of this PowerPoint. So the takeaway message here is you need to understand that enzymes catalyze and speed up chemical reactions. And they do this by lowering activation energy. Okay, now this applies to exergonic and endergonic reactions, but it's much easier to picture this in an exergonic reaction. So I really just talk about exergonic reactions when I teach about enzymes. Now remember that enzymes are specific to certain substrates. So like lactase breaks down lactose. The enzymes are not used up during the reaction, so they can keep on catalyzing reactions. And finally, I want you to remember that enzymes are proteins. Remember, proteins are so important in an organism. They give you all of your characteristics, eye color, hair color, skin color. And they're also super important because they're enzymes. And your genes in your DNA code for proteins. So that is the end of this lecture. The next lecture will be on the cellular energy dynamics part 2 PowerPoint. And for that one, I'm going to go into the process of cellular respiration.